Hello, my name is Yeonggwang Park. I'm a PhD student at Boston University. Today, I'm happy to present our project title, Vocal Fold Kinematics and Relative Fundamental Frequency as a Function of Obstruent Type. Relative Fundamental Frequency, RFF, is an acoustic measure that quantifies instantaneous changes in fundamental frequency during the transition into and out of a voiceless consonant. As you see in this figure, RFF is obtained in a voice sonorant, voiceless consonant, voice sonorant utterance context such as alpha. RFF is calculated by comparing the instantaneous fundamental frequency values of the 10 voicing cycles immediately before and after to the voiceless consonant to steady state values, the fundamental frequency values of offset cycle 1 and onset cycle 10, which are shaded in the figure. These cycles are used as reference cycles, and RFF of each cycle is calculated with this equation. In this way, RFF examines changes in fundamental frequency as a speaker terminates phonation, which is voicing offset, and reinitiates phonation, which is voicing onset. RFF has shown promise as a non-invasive, objective measure for assessing vocal hyperfunction. Vocal hyperfunction is a common feature of voice disorders and is usually described as excessive or imbalanced laryngeal muscle forces in voice production. Previous studies have shown that individuals with vocal hyperfunction, such as polyp, nodules, and muscle tension dysphonia, MTD, they produce lower average RFF values at both voicing offset and onset than those of typical speakers, as you see in this figure adapted from Stepp et al. 2010. Despite the potential for RFF as an objective tool for assessing vocal hyperfunction, the underlying physiological mechanisms are still unclear. This figure presents 3 physiological factors for RFF patterns that have been hypothesized previously. First, increase in vocal fold tension from cricothyroid muscle activation. Second, abduction to devoice before the voiceless obstruent. Third, changes aerodynamic during the voiceless obstruent production. In this study, we examined hypothesis on vocal fold abduction by Watson 1998. The vocal folds abduct to assist in devoicing during voicing offset, and it has been hypothesized to lead to decrease in RFF during voicing offset due to decreases in the vocal fold contact area as the vocal folds pull apart. However, there is no empirical evidence supporting this hypothesis. In this study, we sought to evaluate the hypothesis from Watson 1998 by examining differences in RFF and vocal fold kinematics between 2 intervocalic voiceless obstruents, fricatives and stops. In the following slides, I'm going to summarize previous findings on possible differences in vocal fold kinematics between fricatives and stops that motivated our hypothesis. First, let's compare voicing offset prior to T, a stop, with F, a fricative. Stops require a full oral constriction, unlike fricatives. More complete oral constriction can stop the airflow and thus make devoicing easier for stop production relative to fricative production. Thus, less devoicing strategy may be required in stop production, and the vocal folds may be less abducted during voicing offset for stops relative to fricatives. Less abductive vocal folds have less decreases in vocal fold contact area, which may be related to higher offset RFF for stops relative to fricatives. Now let's compare voicing onset after stops with fricatives. Stops are produced with burst, and the burst can result in instantaneous increase in airflow rate, and previous work has also shown that vowels are produced with higher airflow rates when following a voiceless stop rather than a voiceless fricatives. This increased airflow rate may initiate vocal fold vibration when the vocal folds are farther apart, less adducted, which is similar to the observation in a previous study examining ex vivo human larynges. Less adductive vocal folds have more decreases in vocal fold contact area, which may be related to lower onset RFF for stops relative to fricatives. We also suspected that higher airflow rate from burst in stop production may require stiffer vocal folds in order to maintain airflow rates compatible with typical human phonation. This speculation was based on previous findings of a computational modeling and hook wire electromyography study. And based on the finding of a previous study on RFF and kinematic estimate of laryngeal stiffness, stiffer vocal folds may be related to lower RFF at voicing offset and onset for stops relative to fricatives. So the hypothesis of this study is inside this box. For vocal fold kinematics, we hypothesized that stop would have less abducted, stiffer, and less adductive vocal folds than fricatives. With respect to RFF, we hypothesized that offset RFF would not be different between the two obstruents due to the antagonistic effects of less abducted and stiffer vocal folds. We hypothesized that onset RFF would be lower in stops than in fricatives due to stiffer and less adductive vocal folds acting together to decrease RFF during voicing onset. 20 typical speakers were recruited to participate in this study, and as they repeated the utterances EF and ET with a comfortable voice and equal stresses on both vowels during flexible high-speed video endoscopy, simultaneous HSV and acoustic recordings were collected. A series of vocal fold kinematic measures were extracted semi-automatically from HSV images using a graphical user interface described in Díaz-Cádiz et al. 2019. This algorithm uses differences in pixel intensities between the glottis and the vocal folds to estimate the glottal angle during the vocal fold movements as a function of time. Within the graphical user interface, a single technician examined the glottal angle waveform in order to locate a series of relevant time points necessary to estimate vocal fold kinematic measures of our interest. 2 time points were extracted during devoicing. The first time point was start of abduction, and the technician marked T-abt as the last complete or maximum contact of the vocal folds during the voicing offset. Next was time at voicing offset. T-off was marked as the termination of the last vibratory cycle prior to the obstruent. Glottal angle at the voicing offset was extracted at T-off, and duration of voicing offset was calculated as T-off minus T-abt. 2 time points were identified during voicing onset. The time of voicing onset was marked as a time point in which the vocal folds first started to vibrate after the obstruent. The termination of adduction was marked as a time point in which the first full or maximum vocal fold closure during voicing onset was achieved. Glottal angle at voicing onset was extracted at T-on, and duration of voicing onset was calculated as T-add minus T-on. Maximum glottal angle and maximum adductory velocity during the adductory gesture prior to voicing onset were also obtained. A kinematic estimate of laryngeal stiffness, Ks, was calculated as the ratio of Vmax to theta max. RFF was manually estimated from acoustic recordings, and RFF values from offset cycle 10 and onset cycle 1 values were considered for further examination. This is because they have been shown to reflect the largest differences between individuals with healthy and hyperfunctional voices. Wilcoxon sign rank tests were performed on RFF and vocal fold kinematic measures to evaluate hypothesized differences in these measures between fricatives and stops within participants. Statistical significance was adjusted with Bonferroni correction. The vocal fold kinematic measures, glottal angle and duration of voicing offset, were statistically greater in fricatives relative to stops, and Ks was statistically greater in stops than in fricatives. As hypothesized, offset 10 RFF values were not statistically different between stops and fricatives. These results may be due to the effects of stiffer vocal folds hypothesized to decrease RFF, canceling out the effects of less abductive vocal folds hypothesized to increase RFF at voicing offset prior to stop production. For voicing onset measure, glottal angle at voicing onset was statistically greater in stops than in fricatives, and onset 1 RFF values were statistically lower in stop productions. These results may be due to the effects of stiffer vocal folds and less adductive vocal folds, both of which were hypothesized to decrease RFF at voicing onset after stop production. The less adducted, stiffer vocal folds and lower RFF at voicing onset for stops relative to fricatives in this study were in accordance with hypothesized physiological mechanisms for RFF that decreased vocal fold contact area and increased vocal fold stiffness may decrease RFF. Especially, our findings indirectly supported a prior speculation of the relationship between RFF and degree of vocal fold closure, providing another evidence supporting physiological mechanisms for RFF. Future studies should use computational modeling to directly examine this hypothesis. I want to acknowledge Stepp Lab members and NIDCD for funding this research. And here is their list of the references. Thank you very much.